Okay, so welcome back to this next video in which we are discussing the PI3 kinase AKT mTOR pathway and its role in cancer. Okay, so we've now been through the entire PI3 kinase AKT mTOR pathway, and what we now want to look at is its role in cancer. Okay, so we've discussed how the PI3 kinase AKT mTOR pathway is utterly essential in this initiation of cellular division. Okay, so for a cell that is resting to begin the division process, you need a major epigenetic shift. Okay, and to get this epigenetic shift, you need firstly changes on the level of transcription, and then you also need changes on the level of translation. We've discussed that other pathways, such as the Ras-Raf-MEK-ERK pathway, cause the changes on the level of transcription that need to be caused in order for the cell to divide. And then the PI3 kinase AKT mTOR pathway causes the changes on the level of translation, so that these mRNAs that you're producing, which have just been activated to be produced by, for instance, the Ras-Raf-MEK-ERK pathway, that they can actually be translated into a protein, and therefore they can actually have the effect, okay, in driving the cell into division. Okay, right. So we now understand how important this pathway, how utterly essential this pathway is for a cell to initiate division, basically. Okay, and we now acknowledge that tumor cells must have a way of over-activating this pathway in some way. Okay, and we're now discussing common examples of how tumor cells end up having mutations which allow them to have this pathway over-activated, and therefore to have their translation pathways continuously unblocked, and therefore allowing the translation of these mRNAs associated with proteins that are going to be needed to make the cell divide. Okay, so the first example of a way that we looked at for how cancer cells can overactivate the PI3 kinase AKT mTOR pathway is that you can have gain-of-function mutations in receptor tyrosine kinases. Okay, so these are proto-oncogenes, basically. If you get a gain-of-function mutation in them, they're going to over-promote proliferation. Okay, an example that we gave here was in HER2-positive breast cancer. So in breast cancer, where overproliferation is driven by having too much HER2 receptors on the surface of the cell, and this results because of amplification of HER2 genes. Basically, you end up with too many HER2 genes. These overproduce HER2, and that then overactivates the Ras-Raf-MEK-ERK pathway and the PI3 kinase AKT mTOR pathway, and causes the cell to over-proliferate, basically. Okay, and we've discussed that a key part of why HER2 amplification causes over-proliferation is this PI3 kinase AKT mTOR pathway. Okay, right, and specifically in breast tumours, the PI3 kinase that will be involved in overactivating the PI3 kinase AKT mTOR pathway in response to too high activity in the HER2 receptors is PI3 kinase alpha, since this is the main one that is actually activated by receptor tyrosine kinases and is expressed in tissues other than just leukocytes. Right, okay, so that's one example of how you can overactivate the PI3 kinase AKT mTOR pathway in cancer cells. Okay, other examples are you can have actual mutations in the P110 alpha catalytic subunit of PI3 kinase alpha. Okay, and this itself can lead to overactivation of this kinase, and then it will drive the PI3 kinase AKT mTOR pathway into overdrive, basically, and that can also promote over-division in these cancer cells. Okay, so this is gain-of-function mutations then in the gene for P110 alpha, and remember that gene was called PIK3, okay, CA. Okay, so you can have gain-of-function mutations in PIK3CA, which then overactivate PI3 kinase alpha and overdrive the PI3 kinase AKT mTOR pathway, and this will then mean that you permanently unlock the translation machinery then to these elongation factor 4e sensitive mRNAs, you've permanently got the ability to translate these elongation factor 4e sensitive mRNAs, and this will mean potentially that this way of blocking a cell from dividing that was previously there has now gone, and this might then lead to the cell over dividing. Okay, right. So there are two very famous mutations in PI3K which cause its overactivation and seem to drive cancer cells into overproliferating. Okay, and these are E542K. Okay, E542K, and also E545K. Okay, and these are both examples of missense mutations where a single amino acid in the polypeptide has been replaced with another amino acid. Okay, so to understand this, again, you need to know single letter amino acid code. Okay, so E is the single letter code for glutamate, the amino acid glutamate, whilst K is the single letter code for the amino acid lysine. Okay, so E and K are the two important amino acids in both of these mutations. So in both mutations, what you're doing is turning a glutamate into a lysine residue. Okay, you're turning an acidic residue, which will give away its proton and end up with a negative charge, for a basic residue which will accept a proton and gain a positive charge. So effectively you're turning a negatively charged residue into a positively charged residue. And you'll notice that it's extremely close by that these two mutations can occur. Okay, either you can change the glutamate at position 542 into a lysine, or you can change the glutamate at position 545. So if I draw a little picture of this, let's say this is our P110 alpha. Okay, basically you have two glutamates very close by in the normal P110 alpha. You have a glutamate at position 542, and you also have a glutamate at position 545, and you can turn either of those into lysines, and this can cause the permanent overactivation of P110 alpha. Now, how does this occur? Well, basically, when the P110 alpha has one of these missense mutations in it, it still binds to its P85 type regulatory subunit, but the binding between the two of them become much weaker. Okay, so if I draw this, here's our P110 alpha here. Okay, and here's our P85. Okay, so if the P110 alpha does have one of these missense mutations in it, it will still bind to its P85 regulatory subunit, but the ability of the P85 regulatory subunit to inhibit it will be hugely reduced, and so the P110 alpha remains permanently on, basically. So whenever it just happens to bump into a PIP2 molecule in the membrane, it will now start converting it into PIP3, and this then makes this pathway constitutively on, basically. You don't now need either a receptor tyrosine kinase signal or a G protein coupled receptor signal or a monomeric G protein signal to activate the PI3 kinase anymore. It's permanently on. Okay, now let me just tell you the portion of the catalytic subunit in which that mutation has actually occurred. Basically, both of these mutations, the E542K and the E545K, they occur in this helical domain here, okay? And that results in the interaction that usually occurs between the P85 subunit and the P110 alpha subunit that results in its inhibition being weakened, basically. So although we said that the main binding site between P85 and P110 alpha was the P85 binding domain here, binding to the inter-SH2 domain of the P85 type regulatory subunit. There are more connections than just that. That's the main one, okay? But there are other connections which are involved in the actual inhibition of this subunit by this subunit. Okay, and by mutating those residues in the helical domain, you can weaken the inhibition of P110 alpha by the P85 type regulatory subunits. Okay, and this can result in the P110 alpha being permanently on. Okay, right, so those are other mutations that are commonly found in cancer cells. Okay, so these are quite common, okay, and they also lead to the overactivation of the PI3 kinase AKT mTOR pathway. One of the most common mutations of all that leads to the overactivation of the PI3 kinase AKT mTOR pathway in cancer cells is a loss of function mutation in the tumor suppressor protein PTEN. Okay, so remember PTEN is the phosphatase intensin homolog protein that converts PIP3 back into PIP2. Okay, and if you get loss of function mutations in PTEN, what that's going to lead to is it's going to mean that when you create some PIP3, it's not going to be converted back into PIP2 as well. Okay, now, why would that lead to a problem? It would only lead to a problem if there was some basal activity of PI3 kinase enzymes. Okay, because if P10 was to lose a bit of its function, then okay, it might lead to signals from receptor tyrosine kinases being amplified a little bit, but it wouldn't lead to the pathway being constitutively on unless you completely lost P10 function or there was some basal activity basically. And it turns out there is some basal activity in PI3 kinase enzymes, and the basal activity, the PI3 kinase enzymes that have basal activity seems to mainly be PI3 kinase beta. Okay, so where the catalytic subunit is P110 beta. Okay, so it's thought that the reason that P110 beta is always a little bit turned on, okay, is that the interaction between the P85 regulatory subunit and the P110 beta is a little bit more weak than the interaction between the P85 subunit and the P110 alpha. So that usually when you have a healthy P110 alpha bound to the P85, the P110 alpha is not on at all. Okay, whereas P110 beta, even when it's bound to its P85, is going to be on a little bit basically. Okay, so it has a basal activity all the time, and it's always turning a little bit of PIP2 into PIP3, and usually this doesn't matter because the P10 is always there to convert it back into PIP2, and that doesn't then lead to signaling. However, if you just lose P10, okay, what can now happen is that this basal signaling caused by P110 beta's basal activity can now be amplified to a level where it actually does drive the pathway forward, i.e. you accumulate the PIP3 to a level where it's now capable of resulting in the activation of protein kinase B, and therefore you can get the spontaneous activity of the PI3 kinase AKT mTOR pathway through the loss of function of the phosphatase intensin homolog. And that's an incredibly common mutation that's found in a huge number of cancer cells. However, usually it's not thought that it alone drives the cancer. Okay, usually it's thought that this mutation facilitates other mutations basically. So for instance, if you had an overactivation of a receptor tyrosine kinase and a loss of function of P10, then your receptor tyrosine kinase would drive the production of PIP3, and then that would be amplified by the loss of function of the P10, so the signals would become absolutely huge, and they'd work together to produce total overactivation of the PI3 kinase AKT mTOR pathway. So it's usually thought that the loss of function of P10 is collaborating with other mutations of components of this pathway to produce the overactivation of the PI3 kinase AKT mTOR pathway, rather than it working on its own. Okay, right. Finally, the other major protein in this pathway that can lead to its overactivation, okay, that is found mutated in a huge number of different cancers, are the Ras monomeric G proteins. Okay, these are found mutated in a huge number of different cancers. Okay, and specifically, it's thought that in KRAS positive breast cancer, Okay, so breast cancer that's driven by KRAS this time rather than HER2. Okay, so in breast cancer cells where the overdivision is caused by a gain of function mutation in KRAS that results in too much KRAS activity. Okay, it's thought that the main way that this actually causes overproliferation of the cells is through the activation of the PI3 kinase AKT mTOR pathway alongside the activation of the RAS-RAF-MEK-ERK pathway. Okay, so if you get overactivation of one of the RAS monomeric G proteins, this too can lead to the overactivation of the PI3 kinase AKT mTOR pathway, because remember, RAS in the on state was capable of then activating PI3 kinase alpha, PI3 kinase gamma, and PI3 kinase delta. Of course, in breast tissue, the only one that's going to be important there is PI3 kinase alpha. Okay, so overactivation of RAS is another common way that you can lead to the overactivation of the PI3 kinase AKT mTOR pathway. And there is a lot of interest in whether blocking the activation of the PI3 kinase AKT mTOR pathway by KRAS in this KRAS positive breast cancer will be able to stop the cells from dividing. Okay, so there's a lot of interest in that at the moment. Okay, now, there are other components of the PI3 kinase AKT mTOR pathway which can end up having mutations in them that can drive cancer. Okay, but they're rarer. So, for instance, you can have mutations in protein kinase B that lead to it being all the time on. You can have mutations in mTOR, okay? You can also have mutations in the 4EBPs. So remember, one of the key ways that the PI3 kinase AKT mTOR pathway actually works is by phosphorylating the 4EBPs, which then cleave off the elongation factor 4Es, which then allows the elongation factor 4Es to assist the elongation factor 4E sensitive mRNAs being translated. Okay, if you get loss of function mutations in the 4EBPs, then that does the job for you anyway. That's one of the main ways that the PI3 kinase AKT mTOR pathway allows this epigenetic switch, which is driving division basically. So loss of function in the 4EBPs is another important mutation that is found in some tumours, but the main four are the ones that I've written out in full: the receptor tyrosine kinase overactivation, PI3K mutations, loss of function of PTEN, that's huge, that's found in so many cancers, okay, and also overactivation of RAS, they're very, very important. Okay, right, so we're now going to talk about how potentially inhibiting this pathway, mainly the thing that's being looked at is PI3 kinase inhibitors, but other components of the pathway can be inhibited as well, such as AKT or indeed mTOR. Whether inhibiting this pathway could be helpful in cancer therapy. Okay, and at present, there's only been really one big success story as far as targeting the PI3 kinase AKT mTOR pathway is concerned, and that's idelalisib, okay, which we'll come on to in a moment. Okay, some of the other clinical trials for other drugs that block this pathway have been disappointing. They haven't had as good effect as we thought they would have, and the main reason that we think that they're not having as good effect as we thought they would have is that we don't think we are giving them in high enough doses to have the effect that we would like them to have. Okay, and the reason we can't give them in high enough doses is because of on-target side effects, i.e. the effects of inhibiting these pathways in every cell in the body. It's not good. Okay, this pathway is very, very important in insulin signaling in so many cells, so if you block it, that's going to have consequences for insulin signaling. Okay, so that's one of the major concerns, that we can't give the drugs in high enough doses for them to have the anti-cancer effect we think they ought to have because of the on-target side effects. There's also the concerns with off-target side effects, so the drugs binding to other things that we never even planned them to bind to and then causing side effects. So basically we think the reason it's not working at the moment is because we're not giving the drugs in high enough doses. We still think that the fundamental idea of blocking this pathway is a good way to go. Okay, right. So let me start off by telling you about selective inhibitors of PI3 kinase enzymes. Okay, specifically they're going to be selective inhibitors for the catalytic subunits. So we'll start by talking about selective PI3 kinase alpha inhibitors, then we'll talk about selective PI3 kinase beta inhibitors, and then I'll talk about selective PI3 kinase delta inhibitors, which are used at the moment to treat chronic lymphocytic leukemia. And this example that I've been muttering on about all through the video is idelalisib, and we'll come to that in a moment. Okay, right, so let's start off with selective P110 alpha inhibitors. Okay, so I've got some examples of selective P110 alpha inhibitors, and these are drugs that merely inhibit the P110 alpha catalytic subunit, okay, without inhibiting P110 beta, gamma, and delta. Okay, so obviously we can reduce our on-target side effects by being more selective with which catalytic subunit we're actually going to target, okay, rather than just using a pan PI3 kinase inhibitor which inhibits all the catalytic subunits. Okay, so selective P110 alpha inhibitors, at the moment, because they're not clinically used, they don't have a proper name. Instead, they have code names. Okay, so one example is BYL719. Okay, another notable example is GDC0032. Okay, so those are the two notable examples of P110 alpha inhibitors. Okay, so they selectively inhibit the P110 alpha catalytic subunit and stop it from being able to phosphorylate PIP2 to PIP3. Okay, so there is a lot of hope that these might be useful in treating solid tumours, such as the two that we've mentioned, breast cancer tumours. Okay, so for instance, we mentioned that HER2 positive breast cancer overactivated the PI3 kinase AKT mTOR pathway via P110 alpha, okay, because remember P110 alpha was firstly the only one expressed in breast tissue that could be activated well by receptor tyrosine kinases and RAS, okay. We also talked about how it was the one that would be activated by KRAS positive breast cancer, again because it's the only one that's expressed in breast cancer and which can be activated by RAS monomeric G proteins. Okay, so there's hope that these drugs will be able to be very effective in treating these forms of breast cancer because they will selectively stop the form of the catalytic subunit of PI3 kinase that is responsible for these cells over-dividing, basically. Okay, and if we could cut off the PI3 kinase AKT mTOR pathway in these cells, the idea is then we could stop them over-dividing. Okay, unfortunately, as I say, clinical trials in this are proving disappointing. The effect is very, very modest. It does not seem to have a massive effect. And the problem is, as I've been saying before, on-target side effects. We can't give these drugs in high enough doses, we don't think, for them to actually have the effect that we think they should have. Okay, and the reason is insulin signaling. Insulin uses this pathway, PI3 kinase AKT mTOR pathway, and the main form of PI3 kinase it uses is P110 alpha. Well, the one where you've got P110 alpha as the catalytic subunit, so PI3 kinase alpha. Okay, so these drugs will stop insulin signaling, and therefore we can't give them in too high doses, otherwise we will end up giving you horrendous diabetes, basically, okay, and other side effects beyond just insulin signaling. So that's the main reason we don't think that they're having as big an effect as we think they ought to, because we can't give them in high enough doses because of the on-target side effects that they have. Okay, right. The other hope for selective P110 alpha inhibitors is that we will be able to use them in forms of cancer where you have a mutant form of PI3K-CA. Okay, so where you've got one of those mutations, so remember the two main mutations were E542K and E545K. Okay, so in forms of cancer where you've got a permanently on form of P110 alpha, the idea is that P110 alpha selective inhibitors will be helpful there as well. Again, there's the same problem of not being able to give them in high enough doses for them to actually work. So where we are turning to with regards to these forms of cancer, where you have mutant forms of P110 alpha driving the overdivision, is maybe we could design drugs that would only inhibit these mutant forms of P110 alpha and would not target the normal form of P110 alpha. If we could design drugs that would only target these P110 alphas with these mutations, then we would have a drug that selectively inhibits this enzyme in the cancer cells, and that would be a very, very nice drug, okay, providing, of course, that it didn't have too many off-target side effects, okay? So that's where we're turning to with this. Again, there's the problem with the selective P110 alpha inhibitors. They do work against these mutant forms because, remember, these mutations are in the helical domain, okay, not the catalytic domain. So the catalytic domain still is the same, basically, and therefore these drugs still work, okay, but it has the same problems as we had here, which is that it has too many on-target side effects to be given in a dose where it's actually safe, where it's actually effective and safe at the same time. Okay, right. So that we're turning towards designing drugs that will be specific for the mutant forms of P110 alpha. Okay, right. In cancer, in the very rare instances of cancer where the division is being driven by PTEN deficiency and nothing else, then it would be a good idea to give drugs which inhibit P110 beta selectively. Okay, so remember, if you've only got a mutation in PTEN, the phosphatase intensive homolog, okay, then that can overactivate the PI3 kinase AKT mTOR pathway by amplifying the basal activity of PI3 kinase beta. So remember, PI3 kinase beta is the only one that has a basal activity because the inhibition it gets from the P85 type regulatory subunit is weaker than the inhibition that some of the others get, such as P110 alpha. Okay, so PI3 kinase beta normally turns some PIP2 into PIP3, and if we could block this activity, then it wouldn't matter that you had the PTEN deficiency, basically. Okay, so this is where selective PI110 beta inhibitors are being looked at. Okay, so an example of a selective PI3 kinase beta inhibitor is the drug GSK 2636771. Okay, 2636771. So again, it's got a code name because it's not clinically used yet. So this is an example of a selective P110 beta inhibitor, and it might be useful in forms of cancer where the division, the overdivision of the cancer cells is being driven solely by the PTEN deficiency mutation, the loss of function in PTEN. Okay, right. So now let's turn our attention onto PI3 kinase delta inhibitors, but I think I'll actually do this in the next video, so we'll call it there for this video, and in the next video we'll turn our attention onto idalalazib, which is a selective PI3 kinase delta inhibitor.